Okay, I think we ought to make a start to this afternoon's session, which is following on from the previous speaker. We have 4 more talks looking at getting the right models. The model is incredibly important to getting the molecular replacement to work, not actually for putting the structure in the right place. I remember probably 2 molecular replacement talks at meetings ago, probably about 12 years ago. A colleague of mine presented for fairly convincing work that he could get homology models in the right place in the unit cell. What I then subsequently did some work with him is, certainly using the programs at that point, not many of those would refine fairly automatically. You could always manually build to the right structure, particularly if you knew what the final right structure was. But I suspect things have moved on a lot in 12 years, and what was then probably beyond the radiance of convergence of refinement programs and trying a wide range of models is now much more possible, and I'm sure we're going to hear talks on that this afternoon. So rather than take more of the speaker's time, I'll introduce the first speaker, who is Marco Marcia from Yale University, and he's going to remind us that there's not just proteins in macromolecular crystallography, there are nucleic acids, and you can do quite a lot of clever things with molecular replacement using nucleic acids. So without further ado, I'll pass you over to Marco.
Thank you very much. So thank you for the introduction, and thanks to the organizer for giving me the chance to present this seminar today. It was actually very stimulating to receive this invitation, because it brought us to perform some investigation on molecular replacement of nucleic acid. That is a topic that we use a lot in our lab, but that we realized we didn't know as deeply as I found out in the last couple of months. So actually, the first observation that I made when I received the invitation and started preparing this talk is that when one searches on the PDB, one can notice that there are much fewer nucleic acid structures solved by molecular replacement than protein structures. And so I asked myself whether nucleic acids require to be handled in any different way in respect to molecular replacement strategies than proteins. And if that had to be the case, that had to lie on the differences in the structures of nucleic acids versus proteins. And so as an introduction, I'll briefly touch on those differences. You can see that from the primary structure point of view, for sure, nucleic acids are much simpler than proteins and are composed of fewer building blocks. Also, from the secondary structure point of view, nucleic acids are peculiar because, on one hand, their secondary structure is, again, generally simpler than protein structure is. Nucleic acids are generally composed of quite idealized helical fragments. And at the same time, the secondary structure of nucleic acids can generally be predicted with a quite high degree of confidence on the basis also of biochemical experiment, and not only of computational alignments and searches. And finally, from the tertiary structure point of view, I also want to make 2 considerations. The first impression you get from this slide is certainly that there are much fewer nucleic acid structures than protein structures. But at the same time, I also want to stress the point that nucleic acids are nowadays well known to adopt well-defined tertiary structure motifs, and therefore crystallographic work on nucleic acids is absolutely possible. For sure, the paucity of nucleic acid structures on the databases is a first limitation in the use of molecular replacement on those targets, because with few models, one can also has also got less options to perform molecular replacement searches. However, with my talk, I would like to convince you that this doesn't need to be a limitation for approaching crystallographic studies on RNAs and DNAs molecules, and that indeed, actually, nucleic acids are very amenable to molecular replacement searches, and molecular replacement strategies applied to nucleic acids can even be more flexible and versatile than those applied to proteins. So in general, an approach for molecular replacement work on nucleic acids consists of the following steps. The target has to be crystallized, data has to be collected, and then a suitable model needs to be chosen for performing the molecular replacement search, and the solution needs to be then refined. This is a pipeline that is identical for both proteins and nucleic acids. And also, I need to say that the physics and the experimental setup behind the crystallization and the data collection steps of this pipeline are essentially the same for proteins and nucleic acids. At the same time, nowadays, most software can handle nucleic acids very well, and as well as proteins. So actually, the only point of this pipeline where nucleic acids are significantly more different than proteins is in the choice of the, I believe, is in the choice of the search model, and that's why I'd like to focus my talk on this point. So I'll touch on the following points. First approach, I will discuss with you how to obtain models from available experimental data. And in the second part of my talk, I'll mention some strategies how to obtain suitable molecular replacement search models for nucleic acids, even in the absence of experimental data. And briefly conclude with describing you and reminding you of a tool for model optimization that can increase the chances of success for the molecular replacement search that is being developed in our lab. So one very typical case when molecular replacement is used is when one wants to solve the structure of a target for which there already exists a structure of a homolog. Now, defining homologs for protein structure, we already heard this morning, can follow some well-accepted guidelines. And generally, it is considered that a sequence identity of about 30% or a little bit lower, even in some cases, generally constitutes a good criterion for assuming structural similarity. And the thing is that also true for nucleic acids? And probably for nucleic acids, we have to say that the situation is much more complicated and needs to be discussed to a deeper level of detail. For instance, it is observed that there are RNAs that possess up to higher than 80% sequence identity, but that assume different tertiary folds. And at the same time, there are also many nucleic acids that diverge significantly in sequence, but whose structure, secondary and tertiary structure, are similar. And this is, for instance, the case of the self-cleaving ribozymes, group 2 introns, that I'll mention recurrently during my talk because they constitute the strongest focus of the research in our lab. So certainly these exceptions are also true for protein in some cases, but for nucleic acids they actually constitute rather the rule. These exceptions are understandable if one considers the following observation. Nucleic acids are selected not really for their primary sequence, for their primary structure, but rather for base pair complementarity. And as I mentioned to you, the common secondary structural element of nucleic acids are base paired helices. And then you can easily understand that if evolutionarily one nucleotide on one helical strand varies, and the opposite and the complementary nucleotide varies correspondingly, then a lot of sequence divergency is introduced, but the structural element is maintained. So that's why this covariance model needs to be considered. And again, group 2 introns are a good case to exemplify to you this divergency between primary and tertiary structure in nucleic acids. Here you see an alignment between two group 2 introns that is very poor because it's characterized by the presence of many gaps. but actually by applying the covariance model and by using biochemical data that were collected in a time course of over 20 years, it was actually defined before obtaining the first tertiary structure of a group 2 intron that these molecules are composed and are formed by very similar tertiary structures that are characterized by the presence of 6 major domains, each of which is divided in smaller subdomains for a total of 10 subdomains that here you see highlighted in different colors. And this representation is valid with a certain degree of confidence for all group 2 introns. So, in other words, then the conclusion of this observation is that whereas for protein sequence similarity algorithms are good to identify potential structural homologs that can serve as good search models for potentially good search models for molecular replacement approaches. For nucleic acids, one needs to use rather structural similarity algorithms, and there are a few available already in order to define and to identify potential homologs. At the same time, this difference between protein and nucleic acids leads and raises the following question. Now, those potentially good structural homologs in nucleic acids will then be characterized by very poor sequence conservation. And then, would this poor level of sequence identity be a problem, a limiting factor for the success of molecular replacement searches? So I try to investigate on this point by performing some simple experiments using lysozyme as a test case for a protein structure and the structure of group 2 intron that, as I mentioned, is the focus of the research in our lab and also was my major project in the last 2 years in the lab. So what I did is I took 2 different approaches to determine the dependency of molecular replacement searches on the nucleic acid sequence. On one hand, I generated random sequences for lysozyme and group 2 intron, and I substituted these sequences on the structures of the 2 molecules. Just manually in Qt. These models still maintain a certain degree of sequence identity with the original model. At the same time, I brought this experiment to a further extreme by replacing each single amino acids or base with a completely different one, thereby having no sequence identity in this new artificially created structure in respect to the original model. And for both of these strategies, I used a series of structures to perform molecular replacement searches with increasing levels of RMSD, up to 4 angstroms. And here are the results. So you can see that using the randomized sequences, nucleic acids and group 2 intron in particular behaved much better in the molecular replacement searches, allowing to obtain positive solutions for models that even diverged from the original one to up to 2 angstrom RMSD. And the situation was similar also in the case when I used this so-called opposite sequence that had no sequence identity in respect to the original starting molecule. And instead, the situation was much worse for lysozyme, where I couldn't obtain confidently good solutions. So we can conclude that, that molecular replacement on nucleic acids is definitely less dependent on sequence than for protein. And we can understand this actually easily if we consider that what I already mentioned to you at the beginning, that the building blocks of nucleic acids are simpler than in the case of proteins. And in addition to that, you should also consider that the strongest scattering component of the nucleic acids is composed or is contributed by the sugar phosphate backbone and not by the side chains. I, again, exemplify this observation with a simple test on lysozyme and group 2 intron. And I used an approach that is a little bit reminiscent of the use of polyalanine, polyglycine models for proteins. In the case of group 2 intron, as a search model, I used either the sugar phosphate backbone alone, removing the base moieties, or only the phosphate groups by themselves. In alternative, as a comparison, I tried to use also only the base moieties as search models, and again with series of structures at increasing levels of RMSDs. And what I obtained is that, as expected, the search models containing only the backbone or even the phosphate groups only allowed me to obtain suitable and successful solutions by molecular replacement with corresponding interpretable electron density maps, whereas this was not possible when I used only the bases that contribute to the scattering to a lesser extent. So another standard case when one would use the molecular replacement to solve structure of a target is when the structure of the target is actually already available, but one wants to investigate it also in different conditions. So this approach is sometimes considered problematic for RNA because RNA and nucleic acids in general are considered to be more intrinsically disordered and therefore more sensitive to changes in buffer conditions, ligands, and especially metal ions that are really essential to maintain the tertiary fold and the stability of these molecules. But is this actually true? In my work, I solved a series of 14 different structures of group 2 introns by molecular replacement, and all were solved in very different buffer compositions, and especially in the presence of a wide variety of metal ions. And all of them could be successfully solved by molecular replacement and are highly isomorphous to each other. So this work certainly allowed us to identify a wide variety of metal binding sites and to confirm the identity of a wide variety of metal binding sites in those structures. And to a broader perspective, shows that a wide variety of metal ions, probably wider than previously expected, can support the structure of large RNAs without inducing significant fold changes. This is important on one hand because metal ions are so crucial for large RNAs and very widely used in a number of biochemical and structural techniques. And also because metal ions are very important for the function of these macromolecules. And indeed, the screening of this series of metals enabled me to obtain structures of intermediate stages of catalysis, therefore also giving an insight into the mechanism of action of these group 2 introns, which was not clear on the basis of the first structure that was solved in our group 4 years ago. At the same time, I also forgot to mention that despite this going a little bit far from the major focus of this meeting, this can have also very interesting potential implication for de novo phasing methods because most of these elements are very good anomalous scatterers. And I must say, I was very interested in hearing yesterday about the progresses of long wavelength data collection that may allow not only approaching studies at phosphor using phosphorus anomalous scattering, but also potassium and calcium that are very crucial physiological ions that play an important role in group 2 intron for sure, and possibly in general in large non-coding RNAs. Finally, another approach when one wants to use molecular replacement to solve nucleic acid structures, starting from available experimental data, is when the structure of smaller domains of the target are already available, and one has obtained now a data set for the whole complex or the whole macromolecule. This is a very common case in the protein world. And for instance, here as an example, I mentioned the work done last year by my colleague Dahai in the lab, who solved the structure of RIGI. This is a protein involved in recognition of viral RNA and immune response. To solve the structure, he used the substructures of different domains of the protein as molecular replacement searches, and his work led me to question whether the same would be possible also on group 2 introns that, as I mentioned to you earlier, are indeed composed of 10 different smaller subdomains. And therefore, I broke the structure of group 2 intron apart, and I tried to use these 10 fragments as search models for performing molecular replacement searches on my data. And indeed, to a certain extent of structural flexibility, I could indeed obtain a positive solution and interpretable electron density map for group 2 intron. Now, this result is particularly significant in the light of what another colleague of mine in the lab is doing. Elizabeth is trying to design de novo, or ab initio, structures of small RNA fragments. Her approach is that she starts from structure libraries obtained from the available nucleic acid structures to date. From those, she defines pseudotorsional atoms and libraries of pseudotorsional atoms to guide the assembly of the nucleic acid building blocks. And using such libraries, she can, starting from a given new RNA sequence, for instance, predict the structure, the three-dimensional structure of that particular fragment. And what she obtained, recently published, is that she can correctly rebuild fragments of even up to 174 nucleotide in length with a very good accuracy of less than 1.5 angstrom in RMSD. So those ab initio design fragments can indeed, on the basis of what I showed you before, serve as very suitable and promising models for molecular replacement searches. Especially because not only for RNA it is possible to reach this stage computationally, but also is often the case that biochemical experimental data are available to support the organization of the RNA molecules. It is certainly the case that the smaller molecules, for instance, domain 2, domain 4 in group 2 intron that are characterized by a simpler structure are not so easy to position by the molecular replacement softwares in such complicated searches. And I think that at this stage it is worth mentioning that experimental phasing can, in parallel and in conjunction with molecular replacement approaches, can facilitate the localization of such smaller subdomains. And indeed, in collaboration with Raj, an excellent beamline scientist at APS Chicago, where we collect all of our diffraction data, we tested this, and by using MORREP, we were able to correctly position the domain 2, at least, of group 2 intron into the electron density by using preliminary and insufficiently, not good enough experimental phases. Whereas domain 2 instead, together with domain 4, was often positioned wrong, or the two domains were swapped with each other in the molecular replacement, multiple domain molecular replacement searches that I described to you just now. Finally, I already pointed to you the work of Elizabeth that uses ab initio, or that can design structures of RNA fragments ab initio. And I want to touch more deeply on this topic by showing to you that indeed, even in the absence of experimental data, it is possible to create good models, suitable models for molecular replacement searches in the case of nucleic acids. And besides Elizabeth's work, I'll first describe to you another approach that uses idealized fragments as starting models for molecular replacement. And by describing to you this, I refer to the work in the Scott lab that was able to solve the structure of the L1 ligase ribozyme with the following method. What the authors did in that work was that they defined that that particular target was composed of 4 different helical stems. They predicted this secondary structure, and then they simply designed helical fragments in COOT with idealized geometry. And they inputted these 4 helices as independent search ensembles in phasor, thereby being able to obtain an interpretable electron density map from those data, and experimental interpretable electron density map that was later also very elegantly confirmed experimentally using bromine derivatives and experimental phasing. And finally, also, as we heard already this morning, in the case of protein, also for nucleic acids, homology modeling can be used as a strategy to design and create molecular replacement search models. This is work that in our lab is carried out by Shri, another colleague of mine, that follows the following approach. Starting from sequence alignments, biochemical data, and a known 3D structure, as it can be the one of group 2 intron that we solved several years ago. By using a software called ModeRNA, he can indeed design 3D models of novel targets, of novel group 2 introns, for instance. As a test case here, I exemplify this using instead a simpler molecule, adenine and guanine sensing riboswitches. And what Shri did using ModeRNA is that starting from the structure of one of these riboswitch, he modeled the structure of the other one, and both structures are known. So the comparison of his model with the known structure of that riboswitch shows that the model and the real structures are very close, very similar to each other, and again, in a range of RMSD that, as I showed you in the first part of my talk, is absolutely suitable in the case of nucleic acids to be used for molecular replacement searches. And in conclusion, I just want to remind you of a tool, software developed by Kevin Keating and in the lab, and available now as a plugin in COOT, ArcRain. This software can be very useful for, in general, for modeling and refining manually RNA structures, but in the particular case of optimizing the molecular replacement model, it's again a very useful tool that allows chain extension or deletions and modifications, and even rotamerization and optimization of the rotamers in the structures, both in the presence and for what is more important in this specific seminar, even in the absence of experimental electron density maps. So in conclusion today, I showed you that nucleic acid structure homologs need to be identified using structure-based algorithms and not sequence-based algorithms. However, the advantages that nucleic acids, molecular replacement on nucleic acids is less dependent on sequence. I also showed to you that large RNAs may possess stable, well-folded, and isomorphous structures in a wide variety of experimental conditions. So this can open up future investigations for other large RNAs too. And multiple domain searches are absolutely possible to perform also in the case of nucleic acids, with the advantage here that the structure of the small domains used in the search may be predictable not only on experimental basis, but also ab initio and by de novo design. Again, to a broader extent, homology models and idealized fragments can also constitute examples of search models obtained in the absence of experimental data. So, starting from this consideration, I hope I convinced you that and encouraged you to perform broader studies and to undertake broader studies on large RNAs. Right now, the scenario on the databases is that there are very few nucleic acid structures, wide minority, in respect to instead the situation that is true in the cell, where there is a wide abundance of non-coding transcripts. Most of these non-coding transcripts are now more and more being characterized to possess well-defined tertiary structures, well-defined structural domains. Therefore, I hope that with a little bit of contribution from my seminar, we will in the future revert the tendency and aim at a more equal distribution of known structures of all the different macromolecules. With this, I'd like to end up and just acknowledge all the people that participated in the work. I already mentioned Elizabeth, Kevin, and Shri. Dahai for providing me those snapshots of Rigai and all other members of the PILE lab, and in particular Anna for her support and guidance throughout the project, and in particular also for thorough discussion in the preparation of this seminar. I'd like to thank Raj and all beamline scientists at APS, where we collect our diffraction data, and NIH and HHMI for fundings. You for your attention, and I'm open for questions.
Questions for Marco, and could I ask you if you could give your name and where you come from when you ask a question? Frank.
I'm Frank from Diamond. Is there a metric for homology, if you want? Before, for proteins we've got sequence similarity, and you say you need to use structural searches. Is there some metric that says how likely it is? Because I didn't understand your mutation experiments either, where you said you just change the sequence, but we often can use polyalanine and it will do molecular replacement anyway, so you haven't really changed that experiment, that you have a different fold in there.
Right. So there are different points to this question. The last one, that experiment is just to confirm that that molecular replacement searches for nucleic acids are less dependent on the sequence. So both the use of those sugar phosphate backbones and the use of these mutagenized, artificially mutated structures were both contributing to supporting the idea and the statement that for nucleic acids, the strongest scattering component is contributed by the phosphate and ribose moieties, and that the sequence of the nucleic acids themselves is less important. And I thought this was an interesting point to make, or at least an important point to make, because of what I said just before, that structural homologs of nucleic acids will probably have maybe not in the totality, but in a large majority of cases, would have very different, very far sequence similarity in respect to the target that we are investigating. This brings me to answer the first question you had. I don't know if one can set a cutoff of structural similarity on the basis of structural similarity algorithms and predictions. I'm not aware of this, but certainly at least I can refer to the case of group 2 introns, where if you observe the secondary structure maps of all these molecules in different phylogenetic lines, you can really see a strong covariance and a strong similarity in the organization of the domains and on the long-range tertiary interactions. So this leads you to suppose that the structures are also similar. And yeah.
Yep. Question there.
Hi, over to Southampton. overwhelmingly NMR structures are, nucleic acid structures are NMR structures. So how is the problem therefore determining molecular replacement solution? Has that impacted your statistics that you showed at the beginning?
The statistics refer essentially exclusively to crystallographic data. That's true. So but for sure, NMR data are absolutely, NMR structure are absolutely valid models to be used. I haven't tested them myself. I would expect, yes, absolutely.
Any more questions? If not, I'd like to thank Marco very much. Our second speaker is also called Marco, is Marco Punter from the EBI and Sanger Institute in Hinxton in Cambridge, and he's going to tell us how far we can get with molecular replacement in terms of those families which as yet have no homologous structures. So I'll leave Marco to explain.
This is on? Is it? Can you hear? No? Yes, okay. Okay, so first of all, let me thank the organizers for the invitation. I'm not a crystallographer, and I'm learning a lot. Especially I'm very interested in knowing what are the limits of application of molecular replacement. My goal today is to tell you about protein families and structural folds in the post-genomic era. What I will tell you about is mostly how far are we in covering the protein sequence space with structures. So that would be the main goal of this talk. In fact, I won't talk at all about structural folds. I will talk about protein families for two reasons. One is I work on protein families. And the other reason is that both CAF and SCOP, which are the two major classification databases for protein structure, are in the middle of two major new releases. So SCOP hasn't been released since 2009. A new version, very exciting version, is now essentially ready, but it won't be released until later in the year. And CAF had the release last year, but they're still missing a lot of structures of recent years. So to just go, so for example, if you go to the PDB website, these are new folds in SCOP, new folds in CAF. The blue bars are the new folds per year. And you see that after 2008, 2009, there's nothing. So you could think that, well, you know, we are solving structures, that there are no more new folds. But in fact, the reason is that the databases are not complete. So I hope we will be able to add this analysis to the proceedings if new releases come out of CAF and SCOP. I have to say the statistics here for CAF is for 3.4. There's a new 3.5, but still it's missing a lot of structures of recent years. So, and this is for superfamilies, so you know it's pretty clear that there's something missing. The outline of the talk is very simple. I will start by defining the protein sequence space, telling you what we use as a proxy for the whole protein sequence space. And then I will try to convince you that using protein families, in particular we use PFAM families, are okay for studying the sequence space. So again, the question we have is how far are we in covering the sequence space? So I'll try to show you that PFAM families cover a significant portion of what is relevant of UniProt, of what we use for the sequence space. And then we can use PFAM families to ask the question how many of them are structurally covered. Then I'll go to actually that part. And I want to conclude with a couple of slides on the human protein, which is arguably the most important set, on average at least, not for everybody, but of protein sequences we have. And I want to give you an idea of how far are we in covering that structurally as well. Okay, so protein sequence space, well, by definition is all the sequences, protein sequences, we know. Oh, by the way, I'll talk only about proteins, so we go back to proteins from nucleic acids from the previous talk. We will use UniProt KB, and we use in particularly a snapshot taken in June 2012 that had 23 million sequences. UniProt KB is as good as any other choice of large database sequences. It's the one we use at PFAM. 23 million sequences. Now, the latest release has close to 29 million sequences. About 2-3% of them are in SwissProt. It's a subset of these 23 million sequences. And things that are in SwissProt are reviewed. So that means that there's been a person that has taken one protein or a group of proteins that are homologous and has looked at the evidence for these proteins to exist. It can be expression, can be RNA-seq, it can be homology to other proteins that are known experimentally, and has decided that essentially that's a real protein sequence. Okay? So the others are usually annotated as hypothetical proteins in UniProt KB. Doesn't mean that they're not real proteins, but simply we don't have enough evidence, except for example, annotation from sequencing efforts. I will talk about both coverage of UniProt KB and SwissProt in the following slides. UniProt is increasing very fast. So this is the curve. In 2008, there were about 4 million sequences. Today, there are almost 30 million. And the worrying part of the graph is that it's going vertically, essentially, you see. That's an artifact, of course, but it's increasing fast. Now, the thing we want to do is we have these 23 million sequences. To look at coverage, structural coverage, what we want to do is to cluster them together. In particular, we would like to cluster them into families that we believe have the same structure, right? Because if we have that, then what we can do is we can simply look at the structures where they fall. So, and we can say, for example, oh, there's a structure in this family. Okay, there's a structural representative. We could say this is structurally covered. Of course, this is an oversimplification because large families, it depends what you mean by covered, right? It means that there's one structural representative, but it may not be even good enough to do homology modeling for the whole family. Or for example, studying function, in many cases, this is not good enough. But we will use this oversimplification. We say, okay, so if there's one structural representative, we say that the family is completely covered. Now, how do we get this group of families? Well, what we will use is simply homology. Okay, so we know that homologous proteins, in the vast majority of cases that we know, give similar structure. So we will use homology that can be detected via sequence similarity. So we can do it at the sequence level. Of course, we know that there are proteins that have similar structure and are not homologous, but that's too difficult to do without knowing the structure. So I will use, as I said, PFEM. PFEM is the database I work at, and what PFEM does is essentially to cluster together protein regions that we believe are homologous. One important thing is that we don't cluster full sequences. I mean, sometimes the homologous region is the full sequence if it's a single domain, but we try to group together domains. So we split proteins into conserved regions that correspond to, hopefully, to structural domains. And we do this with quite a number of, quite a few manual intervention. So very briefly, if we have a set of proteins, we identify a set of a region that is conserved. We build a multiple sequence alignment. We curate manually this alignment, taking out some proteins that may be fragments or may be dodgy for some reason. From that, we build an HMM model, which is a statistical model that represents the conservation of this alignment using Hammer 3. And then we have this model that you can run against, for example, your newly sequenced genome and look for further homologs of this family. Okay, so that's the idea. At the moment, we have 15,000 families in PFEM. Again, so this is not an automatic clustering. It has a manual intervention. Now, you may wonder, okay, so 15,000 families, we have 23 million sequences in UniProt. Is this enough to say anything about coverage of the sequence space? Well, I'll try to convince you that these 15,000 families are, in fact, covering a significant portion of what is interesting to us in UniProt. So looking at coverage of PFEM families, structural coverage of PFEM families can give us some indication of how far we are in covering UniProt. So this is coverage of UniProt by PFEM families. On the left side, there's amino acid coverage. This 60% means that essentially 60%, almost 60% of UniProt amino acids are falling into a domain that is found in a PFEM family. Okay? The sequence one means that essentially there are almost 80% of the sequences in UniProt KB that have at least one PFEM assignment. That can be one domain or maybe sometimes a shorter motif. So, of course, there are multi-domain proteins that may have only one or two classified domains in PFEM, and the rest is not classified. There are also regions in proteins, as we know, that are not well conserved, so it would be difficult to cluster them together. The situation for SwissProt is better, is close to 67% probably for amino acids, and for sequences, almost all sequences, except for maybe 5%, have at least one PFEM annotation. I'm going to push it a little bit and ask the question, so if we now consider the portions here of amino acids that are not in PFEM, how much of that is actually predicted to be disordered? So if you use IUPRED for this, of course there are many, many methods, that probably to some extent they overpredict, but you see that for SwissProt, for example, this means that only about 10% of amino acids are not covered and are not disordered. Disordered amino acids, of course, are not to throw away, but for us at PFEM, it's maybe more difficult to find conservation in disordered regions, which are lower complexity, and for structural reasons, I guess, they are less, especially for molecular replacement, of less interest, unless you tell me something different today. Of course, they can be crystallized, can be in complexes, but it's a different story. The little improvement in UniProt KB is due to the fact that UniProt is dominated by prokaryotic sequences, while SwissProt has a lot of eukaryotic sequences that have more disorder. Okay, so I hope I convinced you that it makes sense to use PFEM families that cover a good portion of what is interesting to us in UniProt for looking at structural coverage. The other thing I want to show is that this coverage is not going down because UniProt is increasing very fast. You may think, okay, in 6 months, maybe the coverage of PFEM will be down to 10%. This is not what we're seeing. Most of the new sequences seem to fall into families we have. So, in fact, the coverage adding families has increased slightly over the years. This is size of UniProt more than years. Okay, so now, what is the progress in structural coverage of the sequence space? So the first question we want to ask is, we have 15,000 PFEM families. How many of them have a structure, have a structural representative? The answer is the blue one. So it's 43%. 43% have a PDB representative. 57% have no PDB representative. So it doesn't look that great. So there's more than 50% of the families that we still have to cover. So quite a long way to go. Now, the hope is that, of course, the number of structures, we know that the number of structures per year is going up. This is a graph. It's slightly incomplete, so there's maybe a month missing for 2012, so it will be a bit higher. But, the number of structures, protein structures, here I include also structures that are complexes, protein and nucleic acids, are going up. So the hope is that maybe we are getting better and better in covering new PFEM families. So regions of UniProt that are not homologous to previous structures. What we see is not really the case. Is that it's not really the case. So there was an increase around 2005, a peak that I suspect is due to structural genomics, especially in the US, targeting specifically PFEM families, new PFEM families. But in the last 5 years, definitely this number has gone down. Again, 2012 is slightly incomplete, so it may be at the level of slightly higher than 2011, but still much lower than what we had before. So it looks as if we are solving, although the number of structures is increasing, we are solving less new structures to some extent. And you can see this by just taking the ratio between the number of new families acquiring a structural representative and the number of structures solved. So you see that this is much smoother than the previous one, and only about 2% of the structures solved in 2012 belong to new PFEM families. So for 98% of the structures, there was some homolog in the PDB. Now, one little caveat is PFEM doesn't cover 100% of PDB either. So, and it is slightly dependent, this coverage, on the year. More recent structures are a bit less covered, but it's not that significant. So there's lower than 10% of structures are not in PFEM for each year. And there's a slight dependence on the year. So closer years, especially if you look at things that are, if you remove very small chains and also low resolution things. So you see that there's a little bit more things missing from recent years. But it's a small fraction, so I don't believe this is impacting significantly what I showed before. Okay, so one of the questions that I wanted to ask originally when I started to work on this was, how long before we can have all structural folds, right? But I said folds, unfortunately, we don't have. We don't have recent data. So unless I have a crystal ball, I can't really tell you that. But I can answer an easier question. So from what we've seen, we've seen that only about 2% of structures solved today are new. So I guess I would turn the question around and ask, do we really need to solve new folds? It seems that, by and large, we don't, because we solve structures of proteins that are homologous to things we have already solved. So maybe there's not this big push in getting new folds is not scientifically that relevant. But again, this is an open question. I'm not saying this is the case. But I want to try to elaborate a little bit on why, give you a few ideas of why this is happening. I'm sure you have your own. So the first thing, which is interesting, is I told you that 57% of the families, of the 15,000 PFM families, have no structural representative. But if you look at the domains that are in these families, in fact, only 17% are not covered. The reason for that is that we cover well the large families. So families that have more than 1,000 members, 1,000 domains in different proteins or orthologs in the same protein, or paralogs in the same protein, essentially 75% of these are covered by PDB structures. So the structures we have cover the largest families, while for the smaller families, we have a lot that we still have to cover. But that's a good sign, right? So it means that we structures actually cover a larger portion of the sequence space than the sheer number of families would make you guess. Now, the other thing is that families that we don't cover have a higher proportion of things that are not, have no known function. So in PFM, we have a number of families that we call DUFs, domains of unknown function, for which we haven't found any member that has an annotated function. And you see that in the structures, in the families that have no PDB, 37% of these have no known function, while only 10% have no known function in the ones covered by PDB. And I'm guessing that most of those are from structural genomics. So, there's a good reason maybe not to cover this 37%, that's what I'm saying. The other thing is that things we don't cover structurally are more difficult. So they contain regions that may be complicated. Here I put disorder regions, COI regions, transmembrane helices. And this is the proportion of families with a PDB representative and without a PDB representative that have a significant portion of these regions. And you see that for things that are not in PDB, between 20% to 30% of families have these complicated regions, depending on how restrictive you are, how long these regions are. While for the ones with PDB, we have smaller numbers. Again, it's not that we don't have to solve difficult proteins. Difficult proteins may be very important, but it's just that that's one of the reasons probably why we have less of these. Now, the other thing is, what does structurally covered means really mean? Having one structure for one family may be good for small families, but for larger families may not be enough. So a good example, I think, are GPCRs. GPCR is a large sequence family, with many members. We have already a number of structures, but new structures keep coming out, and every time they come out, they end up in Nature or in Science. And the reason for that is that the functional diversity of GPCRs is very big. So they are all homologous, but they have many different functions that are really relevant to us. So solving the same family, structures in the same family, is not bad at all. It can be perfectly justified. So that's one of the reasons why we do it. Oh, you do it, from saying it in a better way. Now, one question, one just comment I want to do. I've spoken so far about PFM families. Now, what's the relationship really with molecular replacement? So essentially, the question is, if you have a structural representative in a PFM family, can you solve all the other members by molecular replacement? And the answer is probably not. So some families are really quite diverse. Even at 25% sequence identity, you can have quite a number of representatives still. So my guess is that molecular replacement is at the subfamily level. So you need more than one structure to be able to do molecular replacement on the whole family. But there are new methods that are coming out. So in some cases, maybe you can even go higher than the family level. One thing I want to show you is the fraction. So this is similar to what Marco presented before, but it's broken up into years. So it's the percentage of X-ray structures that are not solved via molecular replacement. So there are not solved. Yeah. So there was 100% of the initially, of course, of the structures were done not by molecular replacement, and then over the years this percentage increased. I don't know what this peak is about. I checked. It's what is in the PDB. I don't know. Maybe you have an idea. But so the fact is that now, today, there are about 20% of structures that are not solved by molecular replacement. So if you compare this with the percentage of new families, you see that in principle you should have 2% if one structure in a family would allow you to solve all the members by molecular replacement. Instead, still 20%. For 20%, we can't use molecular replacement. So that tells you that PFAM families are a bit higher up than molecular replacement clusters in terms of diversity. Now, I want to conclude with just a couple of slides on the human proteome, and because again, so this is of course a data set we are very interested in, and it may be interesting to know how good is coverage, structural coverage. So this is the PFAM coverage of Homo sapiens. So this collection, Homo sapiens is in this case the proteins that are in SwissProt, which are about 20,000, the current version of the human proteome in SwissProt. And this is a comparison with coverage in yeast and E. coli. Of course, Homo sapiens has a lot of, at the amino acid level, a lot of predicted disorder regions. Anyway, so this is what we cover. And of what we cover is, accounts for about 5,500 families. Of these, we have a structure for about 55%, a bit better than UniProt, but still 45% of the families that have at least the human protein are not covered. At the domain level, 35% of the domains are not covered, so this is worse actually than what we saw in the whole of UniProt. And in terms of difficult families, there are a lot of them in the families we don't cover in human. So between 30%, the 25% and 45% of the families that we don't cover and that have human representatives have some complicated regions for structural determination. On the other hand, it's hard to argue that these are not relevant. So these are human proteins. Sooner or later, probably we want to have most of them solved. So let me conclude by saying, first, diversity of PDB proteins is declining. Over the years, we've just seen there are less and less new structures for new proteins that are not homologous to proteins that were already in PDB. So the question is, are we going towards molecular replacement for all? Not because we are covering the entire sequence space and entire fold space, but because maybe we have covered the vast majority of the families we are really interested in. There's a large portion of PFAM families that are not covered yet. What's left is, however, enriched in small families depleted in functional annotation, and rich in difficult regions. So they are sort of complicated families, many of them. On the other hand, we saw that 35% of domains in PFAM, of human domains in PFAM, are still uncovered, and again, maybe at some point we'll have to tackle these ones in some way, one way or the other. Maybe not structurally, maybe just the functional characterization. Acknowledgements, I want to thank Jaina. We did this work together, and she's a developer, our developer at PFAM. And then I want to thank the whole team. So the Sanger Institute, I should correct this now, we just moved to the EBI, so we are still for a couple of years Sanger EBI, and then we'll go to the European Bioinformatics Institute. This is our team at the Sanger EBI. Then we have our collaborators at Janilia Farm, Rob Finn, Sean Eddy, who's developing Hammer, and Ericsson Hammer in Stockholm. Thank you for the attention.
Questions for Marco? How many PFAM families are represented by a single, say, cath or SCOP, i.e. where actually the sequence homology isn't picking up the structural homology?
So you're saying how many families have more than one cath representative? Is that what you're saying? Or?
No, how many cath families have more than one PFAM family?
I don't know the answer to that. So what I can tell you is that we have about 500 clans in PFAM. Clans are essentially groups of families that we recognize are evolutionarily related, but for which we can't build a single HMM that captures all the members. So they're too diverse to do that. Sometimes we see it from sequence simply. We see that, starting from different points in the sequence space, we get to a number of common members. But when we try to build a single alignment, that doesn't work. In other situations, most situations I would say, we use structural evidence. So, the clear cases in which we have superfamilies in cath or SCOP that have been shown to have very similar structure and maybe even functional similarities. So in that case, we put the families together in the same clans. We have about 500 of them now, but I don't know the direct answer to your question.
Frank.
That fraction of structures that isn't of sequences that's apparently not covered by structures yet. Do you have any sense of how many of those may end up being truly disordered pieces of amino acids, and how many of them will eventually be covered? For instance, by looking historically, maybe 5 years ago, and how many of the things that were predicted to be disordered back then turned out to be ordered after all?
Well, so the prediction methods for disorder, so you're asking essentially how many things are predicted now to be disordered will turn out to be actually perfectly ordered. Well, there's no better answer that I have than the assessments of prediction methods using all the data. So there is a feeling, I would say, that prediction methods are over-predicting. If you look at, for example, there are different issues because disorder is many different things. You can have flexible loops, you can have fully disordered domains, and depending on what you predict, you can predict different things. So if you take many predictors and you look at the consensus, we did this actually for the human proteome. Depending on how many predictors you take, you will have a hard core of amino acids that is not enormous. It's maybe 10-15% that are predicted by all. That doesn't mean that they are the only ones that are like that. My personal feeling is that we don't know the answer to that. As I said, predictions predict a large number of regions to be disordered, but we'll have to wait and see. Also, I think, but that is more a question to you. I have the feeling that crystallographers tend to shy away a little bit from things that are predicted to be disordered. So it's very difficult to get actually enough data to answer your question, I think.
There's a question there.
Yes, I really want to know what's the difference between the PFAM and the CCD from NCBI.
Well, so PFAM is one of many, actually, protein domain databases. So there's TigerFAM, there's SMART, and they all have some small differences. CCD integrates PFAM families in their database. So they somehow look at our families as well and try to integrate into them. In this sense, they are a bit going in the direction of Interpro, which is a meta database that tries to take domain predictions from many databases and trying to find agreement between them. CCD is not really exactly doing that, but it's doing something similar to what we do. So there are differences, but perfectly good database to work with.
If there are no more questions, I'd like to thank Marco again. So the next two talks are looking at ab initio structure prediction for molecular replacement. And the first of the two speakers is Daniel Rigdon from the University of Liverpool, who's going to talk about his program, AMPLE.
So is that on? Yeah. Okay, well, first thanks, of course, to the organisers for the invitation. It's my first study weekend, and it's a really nice atmosphere. I'm enjoying it. So this is a very long title, but essentially it's a novel source of search models, specifically ensemble search models, to try with molecular replacement if the more conventional techniques have failed. What I'm going to do is give you a brief introduction to ab initio modelling, because I don't think it'll be familiar to many of you. And I'll explain just a few of the reasons why we thought there was mileage in exploring ab initio models as a source of molecular replacement models. The results of a pipeline that we've just really finished and published, which is now available to you in CCP4 as a program called AMPLE. And then as we've been going through the project, there have been a few ramifications, further directions that have really opened up to us. And I'll try and describe briefly some preliminary data that we have showing some promise in some unexpected angles, really. So the best way to introduce ab initio modelling is by comparison with comparative or homology modelling. Everyone will have heard of this. Most people have done it. It's a very well-established routine technique, been around for a long time. The problem with the characteristic limitation of homology modelling is that you need to find a template. That template needs to be available. You need to be able to align it accurately and so on. And so in many cases you have targets that doesn't apply to. You can't find a suitable template. And so what we'd really like, what's been the holy grail for many years, is an ab initio modelling, a kind of black box where you feed in the sequence alone, and out pops some structural information giving you a prediction of the protein structure. And after years of sort of pioneering research, mainly by the Baker Group and their Rosetta package, this is now a reality. It's better understood. It's a usable prospect that you can download and try increasingly easily. The limitation here, I should say straight away, is that it only really works well for smallish proteins, up to 100 or 120 amino acids. Of course, many proteins are bigger than that, but when you consider proteins as domains, then actually this is the most common domain size, although many domains are larger. This is the most common domain size. How does ab initio modelling work? Well, very briefly, what you do is you take your sequence and you divide it up into overlapping chunks. For each of those chunks, you assemble a library of fragments from most likely unrelated proteins in the PDB, which you think are likely to well represent the local structure of that fragment. And that's based on sequence similarity and also predicted secondary structure. Once you have a library of fragments for all of these overlapping chunks, you assemble them using a Monte Carlo algorithm. This isn't actually Rosetta, it's a similar program, but essentially you start with some extended conformation, you choose a random position, swap in one of your fragments, and if the scoring function is improved, you will accept that change. If it goes up, then you occasionally accept it in this Monte Carlo fashion. And what happens in these ideal situations is you swap in many fragments, you reach a compact, stable fold, the protein converges on this structure prediction. One characteristic of ab initio modeling is that you don't make a small number of predictions, you make many predictions, typically hundreds or thousands, and then at this stage the models are only represented as polyalanine. The sequence is present implicitly in the calculations, but there are no physical side chains present, and that means that the scoring function here is not good enough to directly pick out the best models. So instead what you do is apply clustering, and what you hope to see within this large number of models is a subset which are quite similar structurally, and when that subset is a large proportion of your model set, then it's likely to represent a fairly confident prediction of the overall fold of your protein. So clustering is key to ab initio modeling, and some representative of that cluster would be your predicted fold for the protein. More recently, the Baker Group and others have added a second stage where you represent the side chains explicitly, and at vastly more computational cost, you can reach some very impressive all-atom models. But what we're talking about today is using these polyalanine models here, these groups of polyalanine models, and these can be obtained in at most 1 CPU day. So this is accessible, whereas these kind of models are not really accessible to most crystallography or even bioinformatics groups. So what encouraged us to consider mining these models for structures to use as search models for molecular replacement? Well, there are three things, really. One is, like I say, an intrinsic part of the ab initio modeling is producing clusters of similar model structures, and that sort of resonates if you're aware of, well, of course you all are, but the idea of molecular replacement using ensembles. As we've heard this morning, ensembles can be an effective way of combining information from more than one structure. So conceivably, we could use those clusters as ensemble search models. Very importantly, we realized, and other people had noted as well, that when you have a cluster of models, those areas of the chain in which the models are very diverse are actually the areas in which the models are likely to be inaccurate. And so we can actually, by comparing along the chain within a cluster of models, we can actually rationally predict which areas, and I'll show you this later on, are more likely to be accurate, and we're going to keep those, and we can potentially truncate off the bits that we believe to be inaccurate and leave ourselves with something smaller but more accurate. And of course, as we've heard several times, molecular replacement only necessarily requires a reasonably accurate partial model of the core of the protein. And like I said, the fast modeling, ab initio modeling, is working with polyalanine structures only, but that's not really a limitation because, as we've also heard already, side chains are often pruned off in the more difficult cases. So there are three reasons that really encouraged us to look at ab initio modeling as a source of search models. We weren't the only people doing this, of course, but there is quite a distinction between the Baker Group here, who've published these two papers, and our approach on the left, in that they very much favor this approach of throwing a lot of CPU at it and coming up with a single, very accurate, beautiful prediction which is good enough to solve the structure. We prefer to recognize that this amount of CPU is a little bit unrealistic for most settings, and so we were trying to make do with these less accurate polyalanine structures here. And we published a proof of principle a while ago showing that within these sets of models there were sufficiently accurate predictions to solve the structures by molecular replacement. But it's important to emphasize here that this was really just a proof of principle, because in this paper we were actually hand-picking what we knew to be the best models and showing that they were good enough to work. So we were really cheating. What we really want is an automatic way to take a set of models and process them into successful search models, and that's what we've been working on these past couple of years. And we've been producing a pipeline, which I'll show you the results of later on, and benchmarking it against quite a large set of structures, actually, much larger than other papers in the area. Almost 300 different structures, no redundancy here, reasonable resolution, but not high-resolution structures by any means, relatively small, considering that size limitation of ab initio modeling. And of course, when we do the ab initio modeling here, we're discarding any fragments from the protein itself and also from any homologous proteins. And so we're treating each of these cases as if it were an ab initio case, a real-life new fold that we had no information about. And it's just been published quite recently. So what I'm going to do now is scroll you through a quite complicated pipeline and pick out some of the key features. So we start off with the target sequence, FASTA sequence at the top. One sequence produces 1,000 of these models. There's this, the word decoy is just a little bit of jargon from the ab initio field. It just means an ab initio model of this kind. We cluster those, and we take the top 3 clusters of those. This is what a cluster looks like, and you can see it looks completely useless. There is a little bit of common structure, this helix and some strands that you can't see quite as well here, but essentially it's far too diverse to be considered as a search model. For each of these clusters, we take at most 200 of those decoys closest to the centroid, and then we add side chains to them with squirrel. And then there's a very important step using Theseus, which is a maximum likelihood structure superposition program. And the reason that's so important is because the maximum likelihood algorithm is much better than a standard least square superposition for finding shared cores between a set of structures and down-weighting structurally variable regions. And this Theseus program, I'll show you later on, it produces information along the chain showing the variance between the set of models. And like I said already, and I'll show again, that is predictive of the error in the model. So based on the Theseus results, we actually do, for each of these ensembles, we do 20 different truncations. We take the 5% that we consider to be the most inaccurate, throw that away, then the next 5%. And so we end up with 20 or more different truncated ensembles from each of these here. And this is what a truncated ensemble would look like. It's starting to look a little bit more homogeneous, but it's still obviously far too structurally diverse to use as a search model. Because of that, we use max cluster, which has just disappeared off the top, and we generate subclusters within. We select from within this ensemble to generate more structurally homogeneous ensembles like this. At this stage, we have 3 subclusters from each of those ensembles, so we're multiplying up again. We have 180 or so here, and then we treat the side chains in 3 different ways. We either keep all of the side chains, we go back to polyalanine, or we just keep the most reliably positioned side chains. And so for each of these subclusters, we have the 3 options of side chain treatment. Then it all gets fed to Mr. Bump, which manages the molecular replacement through both phaser and molrep. And so this search model here actually gave us, after Shell-X, which is very important, I'll come back to that, gave us a relatively complete structure which is being compared to the deposited structure there. So just a couple of the key points of the methodology on these next few slides. None of that whole complex pipeline would be any use if we weren't being able to somehow pick out the best available models in that set of 1,000. This slide here shows that by choosing the top clusters, we generally get at least some of the best available models to work with. And so actually the most common situation we find ourselves in here is a cluster. The top cluster contains one of the best 10 models in that 1,000. So we've got the clustering does work to some extent to enrich our pool in the best models available. This is that key truncation step here. What you have here, these 2 traces, are the red for one of those clusters that we've taken, one of those ensembles. Here in the red, we're comparing the structural variability between the set of models along the chain. So this is a protein of 60-odd residues, and the C-terminus here is where, within that set of models, we see the most diversity. And what's interesting about that is that in this case, and in many cases, that is predictive of the actual error within that model set versus the native structure. Of course, we wouldn't know the native structure in real life, but for the benchmarking, we know it, and we can see that this region that varies most in the model set is exactly where the models are not well representing the actual native structure at all. So in our truncation protocol, this is obviously the region that would go first based on the red trace, but that would effectively remove the least accurate part of our model set and leave us with something that was more partial but more accurate. Then just a word about assessing the solutions. Obviously, because we're working with known structures, we can see how well the molecular replacement has worked in terms of placing the search model. But we wanted right from the outset to use a more stringent criterion of only considering a success where it could be automatically traced, because like you've heard already many times, you can place the model correctly, but the phase bias and the quality of the data don't allow you to proceed easily to automated refinement. So we could measure the accuracy of the placement with ref origin. Very fortunately for us, around about the time we were doing this work, ShellXE emerged as a really effective way of distinguishing between molecular replacement solutions that were going to work and ones that were going to fail. So you apply quite a quick density modification tracing protocol. I'm sure we'll hear more about that later on. And then you look at this score here, and if the score increases and goes above 25%, particularly when you're getting good long fragments, chain fragments, then you can be confident that the molecular replacement is correct and is soluble. And to demonstrate that further, sometimes ShellX itself was enough to take us to some, very near-native structure. In other cases, where it got stuck, we tried ARP-WARP and Buccaneer, and we show that all of the cases that I'm describing to you as successes could be taken automatically to an R3 of below 0.45. This just shows you graphically all of the ensembles, and we have tens of thousands of ensembles dealt with as search models here, and how the ShellX score very effectively partitions out the successes here from, well, in fact, the majority which are unsuccessful. And so this is a very good blind way of knowing whether your molecular replacement has worked, at least for this kind of search model that we were working with. Oh, lost it, sorry. Oh, God. It's all going so well. Right, so that's enough about the methodology. So we applied the methodology to those 300 cases. A couple of slides about which of those targets worked, which of them didn't. What are the characteristics of the targets that work that you could know in advance in order to know whether it's worth trying your protein with this kind of methodology. This graph shows two of the major characteristics that affect whether a target is likely to fail or succeed, which is the length of the protein and also the secondary structure class. And so we saw a big difference between all alpha proteins, which we were very successful with, about 80%, all beta proteins, which were dreadful, and mixed alpha and beta, which were somewhere in the middle. Protein length also affects your likelihood of success. Up to about 100 residues, you stand around a 50-50 chance of being successful. Above that, all of a sudden, you get much more failure than you do success. The failures are the blue bars here, the blue different shades, the red and the yellow are the successes. But there's actually an interesting exception to that, in that even at the longer lengths here, we persist in getting success with those all alpha proteins. So although we haven't explored this extensively, we think that longer all alpha proteins should be accessible to this method. And we've actually just come across one. We've had success with this cytochrome here, which we believe to be the longest target yet solved using an ab initio model. So alpha helical proteins work especially well and may even go above that 120 residue threshold. As you'd expect, we were more successful with the molecular replacement, where we could see that the target had been better modeled. So a rule of thumb to say that we've got the correct fold is a GDT score of above 40. And so you can see here that for the all alpha proteins, on average, we do get the correct fold. For the all beta proteins, on average, we don't. And that's obviously a large part of why alpha works better than beta in the molecular replacement, just the modeling part. But what was really interesting actually is down here in this graph. These darker colors here are showing you successes and the light colors the failures. So here we have the correct fold with these scores up here, and you can see if you get a very good model, you stand an excellent chance of success. But even below this threshold of 40, we see many successes. And what that means, the way we interpret that, is that our truncation regime is rescuing models that overall have been poorly modeled, but in fact possess some kind of core that does look like the true structure. And so we think that these ones here are showing the importance and the success of that truncation for recovering successful molecular replacement solutions, even when the global fold has not been well modeled. And this just shows you that you can, to some extent, predict whether the molecular replacement is going to work from the results of the modeling. If the cluster is very large, I said that at the beginning, that's an indication in the literature that the modeling has worked well. And so you can see from the size of your cluster when you run the ab initio whether you're likely to be up here, large cluster, high degree of success, or down here where it's likely to fail. So that's the characteristics of targets which influence whether they're likely to succeed or fail. What about the search models themselves? Because you saw at the beginning that pipeline generates a large number of search models for each of the individual targets. And we see a whole range of scenarios here, from some targets which are solved with essentially all of the search models, and then a number of targets where only one particular treatment, one particular way of dealing with side chains, one particular subcluster, only one of those search models was actually successful. This shows you the size of the search model relative to the length of the sequence. So these are complete search models up here, where we haven't applied much truncation at all. These are much more brutally truncated search models. And you can see that AMPL is successful over the whole range here. We go right from 5% of the sequence in that successful search model right up to 95 at the other end. But you can see that there's a kind of sweet spot here, around about 20 to 40% of the search model, and that's where we most commonly find successful molecular replacement with these search models. There's an obvious comparison at the lower end here with Archimboldo, which you'll all be familiar with, where they try and put idealized little elements of secondary structure or little motifs in and work their way up from there. That works in this territory of about 5 to 17%, and we're also successful in that territory there, but you can see that we cover the whole range, and some of our search models are much larger than that. This just illustrates for a couple of specific examples how the truncation works in producing smaller but more accurate search models. So if you look at the red trace, for example, here, you can see that the model as a whole, in other words, 100% of that search model, is terribly inaccurate. This is an RMSD of 10 angstroms, so the whole model is looking rubbish. But based on that kind of rational truncation idea, we can trim off the worst of that error, and by the time we're down to 60% of the model, we're down below 3 angstroms error here, and we start to see successful search ensembles deriving even from these models which are overall pretty dreadful. And we can see a range of successes here from small, very accurate search models up to larger but somewhat less accurate search models. So the truncation is really a key part of the whole exercise. That pipeline I showed you was quite complicated, and so once we had all the data, we went back and wondered if all of those ramifications, all of the different ways of dealing with things were actually necessary. And this tends to suggest that they are. What I'm showing here are what we call singleton solutions, so that's where only one of the search models out of the hundreds that are derived for each of the targets, only one of those search models was successful. And what this analysis shows is that all of those 3 different ways of treating the side chains, which are green, blue, and red, we get singleton solutions for each of those. So in other words, if we'd decided not to try one of those side chain treatment methods, we would have lost at least some of these singleton solutions, and the overall success rate would have gone down. The same actually goes for the subclustering here. These are the 3 different thresholds of subclustering. You can see each of them is well populated with singleton solutions. So if we'd decided to just do 1 and 2, we'd have lost these solutions here. The only thing we did find that we could dispense with was the third cluster. Right at the beginning, when we cluster those models, we tried the top 3 clusters, but we found that the third one added no unique solutions, and so as of now, AMPL is only looking within the top 2 clusters. One very funny thing we noticed as we were analysing these results was that sometimes we could have successful molecular replacement in terms of the shell-X and the later refinement, even from very inaccurate models. So again, what we have here is the size of the model in percentage of the sequence, and we can see for very small models, you do tend to need a pretty accurate search model. That's no surprise. But about 5% of them were successful in this range here, where the search model is out by more than 10 angstroms RMS. Very strange, but what we realized is that these are mainly coiled coil or other long helical targets. And what was happening was things like this, where this structure is a tetramer. You can see the coiled coil. The purple here is the search model and how it was placed for successful solution. And you can see that it's just been placed by chance, where it coincides with part of this long helix and then a little helix in a neighboring asymmetric unit. So there's nothing clever about this search model. It's inaccurate, but in fact, with the power of shell-XC, it's actually good enough to bootstrap your way up to a successful solution. And we're actually wondering recently if generating these kind of models, little helix turn helix with a sort of proper packing, may be a general approach for these coiled cases, which we know from expertise in Liverpool that they are particularly difficult. So we're actually going to explore this systematically within the next few months. This slide might actually be obsolete from what I understand about new develops in phaser, but just to show you and compare with a kind of general feeling in the literature, there's a rule of thumb that a TFZ of less than 5 is, essentially not worth looking at least for our models, which may be atypical and non-representative, but at least for our models, we quite often see success, which is the blue bars here, where the phaser TFZ score was less than 5, even down to 2.3. So again, I say it may be related to the search models, but it does suggest that you might want to explore, with shell-XC being available, you might want to explore lower scoring potential solutions from the molecular replacement. Okay, so I'm running out of time. AMPL, that's how it gets its name. It's not as contrived as some you'll see. It's now available in CCP4 in beta form. It requires installation of several non-CCP4 packages, but we're going to try and work to reduce the number of complications for people who are trying to install it. It's also in the GUI, although the GUI does not access absolutely all of the different options that are available. This is still developing and changing, and so there are still things that you can access from the command line, but not from the GUI. I won't talk about that. There is documentation on the wiki that explains in detail how to run it. Because we're not attempting very clever ab initio modeling, the run times are quite reasonable, so that benchmarking exercise around about 1 CPU day for the whole process, including the modeling, and it can all be speeded up using multiple processors or cores. If I've got time, I just want to mention 3 different directions that have opened up to us, only briefly. These are all data from work in progress. 3 different things that we're trying. We're trying Quark as well as Rosetta. Quark, you may know, is another ab initio modeling program recently out, which appears to perform at least as well as Rosetta and perhaps slightly better. So we want to see if it gives us extra successes compared to Rosetta. We've tried AMPL on NMR structures because you can imagine that NMR ensemble can be treated in a similar way to a cluster of ab initio models, which, they vary in some parts, they're well conserved in others. So we've been looking at AMPL versus NMR structures, and also this missing domain cases where you're not aiming to solve a whole small protein, but perhaps you've positioned one domain and you've got a novel domain that you're trying to solve. Just one slide on each, because I know I'm running out of time. So Quark does give us a few extra successes compared to Rosetta, particularly in this mixed alpha-beta category. This success here is another of those. It's really not much more than chance. What Quark has produced is a beta sheet here, 4 strands. It actually spans 2 asymmetric units of the target structure, in which, well, you can see the strands there. So it's another one of these that's a little bit by chance. But I do think that Quark is probably doing slightly better at constructing plausible beta sheets than Rosetta does, because you don't often see such long, well-formed beta sheets in Rosetta models as you do with Quark. We're quite encouraged and pleased by the results first with NMR structures of AMPL. So here, conveniently, is a paper from 2009 where they produced a tool called FindCore, which was specifically designed to find core structures from within our NMR ensembles and use them for molecular replacement. So we compared AMPL's performance on the structures in that paper, and we find, without any fiddling to AMPL, we actually get the same performance really as FindCore on those test cases. In the paper, they went down to the harder cases, less than 20 to 30% range, but with limited success. Although it's early days, we've benchmarked our AMPL against a bunch of thyroidoxin fold structures, and we seem to have much more success in that low sequence identity, high RMSD territory than FindCore does. So early days, like I say, but I think we've almost stumbled across by accident something that works with NMR structures just as well as this specialized tool that's out there does. And then finally, those situations which many of you have been in, where you have one domain and you're trying to find the other, what you'd expect is that having that information from the first domain should make AMPL work a lot better in the second domain. And indeed, that's what we do find. These are 2 different kinds of 2-domain proteins where the second domain is either an insert or a simple segregated terminal domain. The headline rate seems to be a lot better, as you'd expect, when we use a homologue for the first domain. So we're not using the first domain as we find it in the deposited structure. We're finding a homologue within that 40 to 80% range and positioning that and using that as a starting point to ask AMPL to model and solve the second domain. And essentially it works very well. Points worth noting here, we get some successes above 120 amino acids, so it's quite possible that with the benefit of that first domain in place, that size limitation of 120 amino acids on AMPL actually extends a little bit more, because we already see successes beyond that. And we also, although these numbers are small, we also seem to see a little bit extra success for the insert domains, and that may be related to the extra information we have from them. Where there's an insert domain, we can actually tell Rosetta during the modeling what the approximate spacing of the ends of it. If the insert is here and here's your insert domain, we know roughly how far apart the ends are, and we can give that to the modeling as extra information, which improves the accuracy of the modeling. So, new avenues that we're quite excited about. Just to conclude, so on that small protein set, we get 40-odd% success, doing particularly well with all alpha models. And so I think we can fairly conclude that an intelligent treatment by clustering and truncation of those cheaply obtained polyalanine models is a viable alternative to the highly CPU-intensive all-atom approach. You can predict to some extent whether your case is likely to work with AMPL based on its size and based on its predicted secondary structure. For us, ShellX was an absolute boon in terms of picking out the correct MR solutions. It works wonderfully well. We think we've stumbled across something that works well for NMR models, and the missing domains were even less further in properly characterizing that behavior than with the NMR. And if you're interested in talking to us, having us look at a particularly different case, we're always interested in talking to you. So acknowledgements, this was mainly Jacqueline's work at Liverpool here. Olga is a crystallographer who had lots of interesting ideas from the real sharp end of the user's point of view. Martin and Ronan you'll be very familiar with. Ronan in particular I'd like to credit with bringing lots of interesting case studies from these schools and workshops. He goes around the globe. He's brought back lots of interesting ideas and experience with AMPL. And Yang Zhang kindly calculated and provided us with those quark models. Thank you.
Questions for Daniel? Pietro?
Pietro Bilbao. I'm interested in the dependency of the success or failure on the quality of those initial Rosetta fragments. Because you use those fragments at the beginning, don't you?
The quality of the fragments?
Yes, because they are fetched from a database of existing structures, basically, looking at structures of the sequence. How close to the actual structure are those?
Well, those fragments are only sort of 3 to 9 amino acids long. So, you know, even within unrelated proteins, you're very likely to find a chunk that matches almost perfectly that unknown region of your protein.
Yes, but when it fails, and you go back and look at the ones that were presented, do they match?
There doesn't seem to be much feeling that the fragments is a limitation. You know, there's the big world of protein structures to play with, and you know, from reading the literature, I don't think that's generally considered to be one of the limitations. So.
Yeah, on the front row there.
I was wondering, out of the many success cases that you've shown, how many of those proteins are oligomeric proteins?
Oligomeric, so
I think that's an interesting statistic in the general field.
Yeah, it is. I haven't got those statistics here. I'm even wondering if they're in the paper now. Certainly many of them have 2 or 3 subunits in the asymmetric unit. So the failures are certainly not due to, you know, it being one subunit among several. That doesn't... I don't have strong statistics, but I don't think that's an issue generally.
Frank?
Do you have a sense, or did you ever try building a small domain into a larger protein where, say, the small domain is quite alpha helical, for instance, and only 30% of the whole thing, and then using that to bootstrap the whole structure?
Well, I mean, that's what we're doing a little bit at the end with those domains, no?
The other way around. I mean the other way around. The first domain you're looking for is one that you can
I see, right.
reliably, but see if that will help you.
So where we tried to build only part of it, no, I don't think we've done that, no. No. There's many things to try.
Okay, if there's no more questions, I'd like to thank Daniel once again. And the final speaker of this session is Frank DiMeo from Ted Baker's group in the University of Washington. At least I'm guessing it's Ted Baker's group from the University of Washington. And he's going to be talking about molecular replacement in Rosetta.
All right, can everyone hear me okay? All right, excellent. So yeah, I'm Frank DiMeo from David Baker's group, and I'm going to be talking about the use of Rosetta structure prediction to solve difficult molecular replacement problems. Sorry, let me just adjust this a little bit. All right, so what makes molecular... All right, sorry about that. Anyway, all right, so what makes molecular replacement problems difficult is when the template structure that we use for search is very distant from the target structure. And when molecular replacement fails, there's really two places that it can fail. So first of all, oftentimes the template itself is good enough to place the molecule in the unit cell, but the resulting density map is uninterpretable. And so I'm going to show how we can use Rosetta structure prediction guided by experimental data in order to rescue these difficult cases. And then also I'm going to talk about some more recent work where in really difficult cases where we're not even able to, so we can identify template structure, but we're unable to actually even place that into the unit cell. And I'll talk about some advances that we've made in homology modeling in Rosetta in the past 6 months or so that may allow us, in some cases, to rescue these difficult cases and to place the molecular replacement model. And so basically then my talk will discuss these two different approaches. First of all, using weak experimental data in order to guide refinement of these molecular replacement solutions, and also these methods for improved homology modeling. And so I'm actually going to start with just a brief introduction to what Rosetta is and how we can do structure modeling in Rosetta. And so Rosetta is a protein structure modeling package. It's been developed by the Baker Lab over the past 15 years. You actually heard a little bit about it in the last talk. And it includes software for both ab initio structure prediction, homology modeling, protein docking, and even protein design, where we try to design a sequence that will adopt a particular fold or perform a particular function. And so even though these tasks are all quite different, really the key thing that tie them together are the fact that these protocols all draw off of knowledge of biochemistry as well as information determined from previously solved protein structures in order to define an energy function. And this energy function describes, given a particular protein conformation, how likely it is how native-like this protein conformation is, and also ways of sampling conformational space. So the conformational space of proteins is huge, and Rosetta includes ways of intelligently sampling these that basically try to find the conformation that globally minimizes this energy function. Really quickly, to introduce the energy function used by Rosetta. So it consists of a number of both statistical and physical chemical terms. And these things include both backbone torsion and side chain torsion probability distributions, as well as things like van der Waals energy, implicit salvation, and hydrogen bonding energies. And so finally, Rosetta, so the majority of Rosetta protocols operate in two different phases. In the first phase, known as the low resolution phase, and again, this was also talked about in the last talk briefly, but basically the side chains are modeled using a single interaction center. It's a very coarse-grained energy function, but it's very, very quick to compute with, and it's less discriminative. And so generally, there's sort of large-scale backbone rearrangements are made in the centroid mode. And then the high-resolution energy, this is all the atoms, including hydrogen atoms, are modeled in the protein. It's slower to compute, but it's much more discriminative. And so then generally, refinement is carried out in this high-resolution energy function. All right, so getting back to molecular replacement, like I mentioned at the beginning of my talk, really, molecular replacement becomes more and more difficult as the template structure gets further and further from the target. And so what you see is the same data set here, 1.8 angstrom data set, that's phased using two different molecular replacement models. On the left, we have a very good 50% sequence identity model. You can see the density map is straightforward to interpret. Everything looks really, really nice. On the other hand, if we use this 18% sequence identity model, you can see that it's extremely difficult to interpret. The backbone density isn't visible. It would be very difficult to try to build a model into this density map. And so the way we use, but we can use Rosetta to try to rescue these cases. And so the basic idea is that we perform our initial molecular replacement search. And this consists of not only multiple template structures, but also multiple orientations of the same template structure. And so we find a number of different molecular replacement solutions. One or more of these may be correct. We're not even sure from the phasor z-score or likelihood if there's a correct solution among these. And if there is a correct solution, which one is correct. And so our task is to identify the correct molecule from among all these different candidate structures. And we want to also be able to improve this model so that then if we rephase the data, the resulting map can be interpreted straightforwardly. And so the way we do this is as follows. So for each of these solutions, we assume that they're correct. We use them to phase the data, compute this putative density map. Of course, for all the solutions that are incorrect, this map will be junk. But for the correct solution, although it'll be really noisy, it at least contains some information. And then we perform rebuilding and refinement in Rosetta, and I'll talk about that in a second. But we perform rebuilding and refinement of these template structures into the corresponding density maps. We get new models fit to the density data, and we rescore them against the experimental data, against the unphased crystallographic data. And so basically the idea is that only for the correct solution does the density map signal and the energy signal from Rosetta agree, and we get this reinforcement. So this should be able to easily identify the correct solution, if there is one, among this set. And it also should improve the model enough that we can perform, that we can then, that automatic chain tracing can solve the structure. Okay, so then this rebuilding and refinement step in Rosetta, just to very briefly introduce it, the basic idea is that we want to rebuild get both gaps in the alignment as well as portions of the backbone that poorly match the density or that clashed with crystal contacts. And so the way this rebuilding is done is actually in a two-step process. First, the centroid step, we have some template structure and we have a gap that we want to rebuild. So given the sequence, we can use basically find backbones from previously solved structures that have similar local sequences. We can steal the backbone torsions from those structures and insert them into this segment. The movement of this, when we insert this fragment, we can actually propagate this movement towards the middle of the segment, and this kind of will tear open the segment. But then we can use a geometric closure routine, like cyclic coordinate descent, in order to get kind of a closed-loop conformation that locally agrees with the fragment set. And so you can see an example. And so then each of these, and so this can be done very, very quickly to generate many different closed-loop conformations. And each of these are evaluated with Rosetta's low-resolution energy function together with the experimental density data. Then in the next phase, we go on to the full atom modeling. And so here, basically, we just iterate between two different stages. So here we place all the protein side chain atoms on. And we just iterate between cycles of discrete rotamer optimization. So here, given the backbone, we optimize the side chain rotamers. And this is done with just a long simulated annealing trajectory. And then, given this now backbone and side chain placements, we do minimization against Rosetta's energy function. And here we allow both the side chain and backbone torsion angles to minimize. And so this minimization is in torsion space. And then we iterate this, and the basic idea behind the iteration is that sort of the backbone movement in the minimization may allow for alternate and for better core packing arrangements during the side chain optimization. And so you can see I just, from one starting model, generate 5 different sort of refined all-atom refined models, and you can see that they are, I mean, this is just fine refinement. As in the previous step, we're using the density information to guide us. And so let me just really quickly walk through an example of this. So this is from the MR Rosetta paper, and this is this XMRV protease. So for this, we found the molecular replacement solution. However, automatic chain tracing with Phoenix Auto Build failed to produce a solution below, I think, 52% are free. And so basically, the molecular replacement solution is shown in blue, and the corresponding density map is shown here. And you see the density is not very clear initially. But by basically rebuilding gaps in the alignment and then refining this with Rosetta's all-atom energy function, we're able to improve the model, and then the corresponding density map looks significantly better. At this point, then, automatic chain tracing with Phoenix Auto Build was able to get a solution that the crystallographer was basically at completion. The crystallographer was easily able to take this to completion. And so, in fact, at this point, we wanted to see basically how applicable this method is. How often do we find molecular replacement solutions that we're unable to take to completion? And so we contacted crystallographers and asked if they had structures that seemed suitable for molecular replacement, but that they were unable to solve. And so we got 18 of these data sets. We then tested all of them with MR Rosetta. And of those 18, 13 of them we were able to solve. And so here we're considering so let me explain. So these are the 13 structures that we solved. And the y-axis is just R-free. The Rosetta-refined structures are shown in green here. And I'm considering a structure solved if R-free is below 40%. So then, after we'd solved these 13, we wanted to say, was Rosetta MR necessary to solve this? And so we went back and we looked with several other methods. Using the template that was ultimately used to solve the structure, we went back and said, well, could we have solved this structure using these alternate methods? And so it turns out for the first 5 of these cases, it actually was solvable by other methods. But for the remaining 8, only MR Rosetta enabled solution of these structures. Additionally, we ran this on a fairly large benchmark set of 59 different